It is your responsibility to check the cleanliness of your objective before imaging. The numerical aperture of a microscope objective is a measure of its ability to gather light and resolve fine specimen detail at a fixed object distance. Low magnification objectives such as 4x, 10x, 20x are generally dry objectives that allow you to easily scan across the sample because they allow a big field of view and thicker optical sections. You do not want to get any oil on these objectives, as it will affect your ability to obtain an image. You may need to flip out the front lens of the condenser for the 4x depending on the microscope and condenser model. Otherwise, your eyepiece field of view will be limited. High-resolution objectives such as 40x, 60x, and 100x are usually immersion objectives for refractive index matching, giving us a numerical aperture above 1. This gives us nice resolution, but it comes at the cost of having a really small field of view. Often you can't do big tiles because the oil would run thin as it moves across the slide. Utilizing a medium with the same refractive index as either the sample or the cover slip reduces refractive index changes between interfaces of sample and chamber, and thus the light is optimally transmitted to the detectors. Therefore, it is important to use the correct immersion medium, whether it be water, glycerol, silicone or oil for that particular lens and sample combination. The correct immersion fluids will be supplied along with lens cleaning tissue with the microscope. Speak to staff if you have any concerns. To prepare for imaging, clean all the objectives. Take a piece of lens tissue from the box and only touch the tissue along the edge, being careful to not transfer oils from your finger onto parts of the tissue that will be used to clean the objective. Fold the tissue in half. Fold the tissue in half again. Turn the turret so the objective is accessible on the side and pushing gently to conform your finger to the lens. Wipe in one direction. It is important to not wipe back and forth, as any dirt on the objective could potentially scratch the lens. Check that the tissue does not have any oil residue. If it does, get a new piece of lens tissue and repeat the cleaning process. It should not have any oil after the second pass, but if it does, get a new tissue and repeat the process. Oil objectives are often spring-loaded, so don't be alarmed if the nose of the objective moves. When you have finished imaging, withdraw the objective from the slide or specimen by using the Z position knob. Remove the slide or specimen and clean the cover slip with a Kim wipe and 80% ethanol. Do not use ethanol or other solvents when cleaning our objectives before or after use. Be considerate and throw all tissues in the bin.